It is my distinct pleasure to introduce you to Dr. Laura Perriman, who is a dear, dear friend of mine and truly an inspiration and a role model. She's a board-certified ophthalmologist, fellowship-trained cornea and refractive surgeon, and ocular surface disease expert. She has numerous publications, has written extensively on ocular surface disease. Her unique and passionate perspective, which you'll get a glimpse of today, on OSD stems from her work in immunopathophysiology. I've been at advisory board meetings, and we're mesmerized by how much she knows about ocular surface disease. She's truly an innovator and enthusiastic speaker who loves bridging the gap between basic science, clinical practice, and patient compassion. So I thank you for being here, Laura, and I look forward to your talk.
Outstanding. Thank you. Thanks, everybody. Really appreciate the opportunity to be here. Thank you for the invitation. I took the assignment seriously. Controversies in cosmetics. Now, men, before you tune out, you need to pay attention to this too, because guess what? Your female patients and your male patients who wear makeup, I don't judge, are going to be asking you questions about what is safe to use cosmetically in and around their eyes. So I want to talk about a few controversies and things that we take as dictum, as wisdom, that we repeat amongst each other that may or may not be true, right? So these are myth busters. So I'm going to start back in time. To cure their pneumonia, just give them a good bloodletting. That's a myth that we perpetuated for a long time until science showed us otherwise. Oh, there's a little microorganism. Great, now we need antibiotics. Here's another one. Vagus nerve, or ulcers are just from stress. Just cut the vagus nerve and it'll be fine. We used to do that to people, right? Before proton inhibitors for severe peptic ulcer disease. And that's not what it is. It's a bacterial infection of the gastrointestinal lining, H. pylori. Here's another one. It is malpractice to put a phaco needle inside the eye. That was 40 years ago, right? 35, 40 years ago. And what do we know now? It's the safest, most elegant surgery that we do. Here's another one. Parabens are bad for you. They're found in blueberries, right? So the point of this lead-in is question everything that you're told, right? We repeat these things that we learned in training or that we think we know, that we've heard before, and there's a confirmation bias that happens. But always ask yourself the question, what does the science say, right? And so that's what this talk is all about. I'm going to go kind of fast and furious, so buckle up, because I want to leave enough time for question and answer. You can carry on the conversation with me afterward on Instagram. Please don't email me. I cannot keep up with it. But you can message me on Instagram, Dry Eye Master. So let's get into it. The controversies. I took my assignment seriously. Okay, here are my financial disclosures. So how do we combat myths, right? How do we combat pseudoscience? Well, pseudoscience starts in a certain way. And you can recognize it by a couple of hallmarks. It's hostile to criticism. Real science, it's like, oh, tell me more about that. Or can you show me more information? Where's the data? Let's study that, right? There's critical difference there. So watch out for that little hallmark. They use vague insights and fear-mongering to create buy-in. Really watch out for the fear-mongering. Fear-mongering amongst our patients is very deleterious to them. They're already anxious about their vision. The last thing they need is fear-mongering, which incites yet more anxiety. They use methods that look a little sketch, right? Watch for that as well. Broad generalizations, watch out for that. Posing as scientifically authentic information. Watch out for all these things and just continue to ask yourself at every turn, what does the science say? I need to read it for myself. Don't believe anything that you're told, even coming from me, because I'm still learning too. So learn to recognize all these things. So, and with that, I'm going to talk about particles and various things in cosmetics. Here is a case report of nylon fibers lodged in the conjunctiva. These had to be surgically removed from a patient. And it got me to thinking, fibers. Here's a case that was discussed on Facebook amongst a physician group. This woman's like, I'm internal medicine, but there's something wrong with my eye. I'm like, what happened? What are you using? Well, she's using this mascara with fibers in it, right? Watch out for these fibers. They're sharp as a knife. The carbon black is also something that's commonly demonized inappropriately. It has been extensively studied by the FDA as a safe colorant, but you hear all the time, ooh, carbon black is bad. Really? Well, what does the science say about it? Well, it turns out that it is toxic if you inhale it, but at low concentrations, i.e. 10%, no more than 10% in a cosmetic colorant, it is safe. So watch out where these opinions are coming from. Really ask that question. I wanted to know how many particles are in our mascaras, and so I did a little study. I looked at 14 different mascaras, and I looked at them under the microscope, and I wanted to know a variety of things. And I presented this at ASCRS last year. So what I did is I taped every single mascara, so I didn't know what it was. Or I had my fellow do this. I was like, I don't want to know what it is. I just want to do the experiment. And then took glass slides, pump, pump, smear 3 times, pump, pump, smear 3 times, pump, pump, smear it 3 times in an artificial tear. So I was trying to test how fast does it dry. Had trouble coming up with that assay. How well does it hold up to an artificial tear and directly suspended? And the middle one was, after it was dried, I tried to swirl in gently without touching or rubbing it, an artificial tear, because I wanted to know how well does it stand up to an artificial tear, as a way of potentially gaining some science around recommending mascara products to our patients. I was shocked to see the number of mascaras that contained these nylon fibers and rayon fibers. Commercial grade, luxury grade, ones marketed to sensitive eye patients have these fibers, and I don't think that they should be there because they do look quite sharp under scanning electron microscopy, and we do have some published literature demonstrating its potential complications in patients, especially the dry eye patient. If you don't have the tear film to flush away those fibers, you have the risk of blinking and those little sharp bands lodging in the conjunctivis. So I really think we need to be avoiding those things. What about preservatives? This gets a bad name. Parabens are bad. Are they? Right? So we actually need to preserve our cosmetics. You don't want mold. You don't want a bacillus infection of the cornea from unpreserved cosmetics products. They have an important role. But how are these things tested? They do have an important role. Their chemistry matters, the biochemistry matters, and the study design matters. So in the case of parabens, I think some of that fear-mongering comes from that one paper showing that parabens were found in breast cancer tissue. There was no control. There was no other metrics. This was one paper, and it sent a fear of panic in the breast cancer community. I'm a fellow survivor. And I'm like, is there something actually to this? Turns out that paper has since been retracted because of its scientific inauthenticity. So watch out for these issues. Parabens are bad in your makeup. Well, if you actually look at cell culture data at low concentrations, parabens do not increase cell death. Here's the control. Here's phenoxyethanol. Here's methylparaben. Here's ethylparaben. At low concentrations, there's no difference over the control, right? So is it harmful? I don't think so. Formaldehyde donors, watch out for this one. Hydroxymethylglycinate. That's a mouthful, right? The instant it comes in contact with water, you have formaldehyde that's being released. Is formaldehyde bad for ocular surface tissues? Yeah. It's irritating to the corneal nerves at concentrations below which you can smell with your nose, 0.5 parts per million. So yes, I do think formaldehyde-donating preservatives do pose a problem for cell cultures. Do we know that it causes a problem for live humans for topical use? We don't know. We don't have those kinds of studies. We're extrapolating cell culture data inappropriately to human in vivo situations. Alcohol, it's 5 o'clock somewhere. But if you have an OH group at the end of a molecule, you can call it an alcohol. So there's another myth. Alcohol is bad in cosmetics. Well, what are you talking about? Are you talking about phenoxyethanol, this common preservative? There it is. Or are you talking about this guy? This is a PABA. This is a paraben, right? They have an important role. Phenoxyethanol is a common paraben alternative. It's commonly mixed in a 2-to-1 ratio with ethyl hexylglycerin as a cosmetic preservative system. And at low concentrations, they are synergistic with each other. So again, going back to the science, is it actually the bad guy? You hear this all the time, avoid phenoxyethanol, avoid parabens. No, avoid a corneal infection from unpreserved products. That's good advice. Here it is again at low concentrations. This is a 24-hour exposure in direct cell culture. There's no way to get mascara inside your meibomian glands where the acini are, where the stem cells are, in order to create anything close to an in vivo type assay, right? These are wash-off formulas, low concentrations. At low concentration, there's no difference over death from the control. Prostaglandin analogs, they are our friend when it comes to glaucoma management. They are our enemy when it comes to jacking up the eyelashes to artificial lengths, okay? So prostaglandin, we know that it's associated with MGD, orbital fat atrophy. This is well commonly described. I remember the first time I saw a lash boost patient. Came in with horrible dry eye, and I'm looking at it and I'm like, why is there orbital fat atrophy? Why is there hyperpigmentation? Why are your eyelid margins so darn red? And why do your eyelashes look like snuffleupagus? Well, it's because the lash boost was laced with a prostaglandin analog called isopropylchlorprostinate. That was the medical expert on that class action lawsuit for failure to disclose the risks associated with isopropylchlorprostinate. So how do you recognize a prostaglandin? Anytime you read those ingredients carefully, anytime you see the root word, prost, like a Polish drinking chair, prost, right? You know that you're dealing with a prostaglandin. So look for these things. M2 eyelash serum has probably pretty high levels. It's popular in Europe. This young woman has horrible dry eye disease, MGD, but from using this product. So watch out for these things. And it was marketed as prostaglandin free. Oh my gosh. You can't even believe the ingredients labels. Lash Envy was pulled from the market in Australia and Canada because they did not put isopropylchlorprostinate on the label. So you can't even rely upon your ingredients labels to make sure your patient isn't being inappropriately exposed to this ocular surface disease exacerbating chemical. I did the, I'm a molecular immunobiologist, like kind of a nerd, and I was able to go through and look at the structure of prostaglandin analogs, and it's basically molecular Legos. You can stick a phenyl group on it, a carboxyl group on it, an ester group on it. You can put some halogenation on the end of it to enhance the potency and the effect, right? That's how we got our glaucoma medications. Turns out, isopropylchlorprostinate here, because of its structure, has the same potency as travoprost, our most potent glaucoma prostaglandin analog. But here's the kicker. They don't disclose the concentration on the ingredients list of these over-the-counter eyelash growth serums. I know what it is. I'm under legal obligation to never disclose it. Tea tree oil. Tea tree oil gets a bad rep too, right? The phyto extracts. It turns out 4-terpenol is important for mites, Demodex mites, and for bacterial control. And you have to use tea tree oil at high concentrations for a prolonged period of time to make any kind of impact on Demodex death. Demodex are like the cockroaches of the ocular surface. They're really hard to get rid of. Soon that'll be different. We've got a medication coming in development. I cannot wait. We're a phase 3 study site for that drug, and that one's called Lotilaner. That's the name of the drug, and you can remember that as a Norwegian drinking chair. We talked about Polish drinking chair, Prost. This one's Norwegian. Lotilaner, yeah, sure, right? Anybody heard Norwegian? All right, anyway. Norwegian Independence Day is May 17th. It's a good party in Seattle in the Ballard neighborhood. Okay, so T4 is anywhere between 30 to 40% the concentration of total tea tree oil, and that's the part that kills the Demodex, controls the bacteria. Most commercial preparations are between 0.1 and 0.2%. Great for bacterial control. Demodex are laughing at you, right? They're like, ha, 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 that's not even close. Yeah, so it gets a bad rap, but the actual concentrations in commercial preparations are quite low and not unlikely to be harmful. Retinols. Who here has heard retinols are bad in your cosmetics? Raise your hand. Yeah? Retinols. That's like saying carrots are bad for you. Carrots have beta-carotene. That's a retinol, right? So it depends on what metabolite you're talking about. Are you talking about 13-cis? Well, if you expose that to meibomian gland cell cultures, you get death. But if you put it on the ocular surface in the tear film, you enhance ocular surface homeostasis and health. That bottom reference should be on the next slide. Oh, there it is. That reference right there, Journal of Molecular Science. Retinoic acid receptors in the naturally metabolized forms are helpful for ocular surface healing. This is one of the reasons why we really like vitamin A containing nighttime ointment, right? To help enhance ocular surface health. Super cool. A lot of words. Here's the point. Cosmetic performance claims, dudes, it is not science. It is marketing research. Let me make that very clear. So if you see some kind of marketing claim that says, oh, 96% of participants reported that their eyelashes look thicker and longer, dude, that's what a mascara does, right? And these surveys are based on Unsatisfied, very satisfied, 0 to 5. If you're marked as a neutral, somewhat satisfied or very satisfied, you're lumped into that 96% category. So be careful of every cosmetic claim that you see. It's not real science. We're used to seeing clinical trials on FDA-approved drugs. This is not that, okay? So be careful of everything that you see. Watch out for those claims. So we've talked about the particles in our makeup, the preservatives and their importance, the safety of them, prostaglandin analogs, and tea tree oil. Pages and pages of references. Happy to talk to you about it. And I finished with 30 seconds shy of 5 minutes, Netta, so we can open it up for questions.
That is a fantastic talk, Laura. Every time you give that talk, though, and I'm really happy to see that you're wearing makeup, because the first time I heard you give the talk, I went 2 days without makeup because you scared me so much. Yeah. And then if you remember, I called you. I said, just list what I can and cannot wear.
Yes.
So
I've learned a lot since then.
Yes, you have.
So I think
we're so excited you were here giving this talk. I think the important thing, and you and I have talked about this a lot, is that as eye care specialists, patients ask us, and maybe us women more than, I don't think Dr. Maloney gets asked what mascara to wear.
He just doesn't know what to say.
So, the onus is on us to give guidance, and there's so much information, it's overwhelming, but thank you for doing all the work you do and educating us about it. I'm still, what I often tell the patients is just kind of follow how your eyes feel, in a sense, for now, and then I'll guide them to you.
Yep.
Let's see, questions. Let's see, do you have any brand of mascara you recommend based on your research?
Oh gosh, I was afraid that one was going to come up. I'm reluctant to name a brand, and here's why. They can change ingredients at the drop of a hat. What was tolerable can no longer be tolerable for your patient. So I'm really reluctant to name a brand, and I apologize for that obsequiousness. Part of it is out of being honest and authentic, and the other part is things can change. So what works now may not work later.
How about, actually, I have a question. Patients often ask, like, how often should I change my mascara?
Ooh, that's a good one. So Demodex love to colonize mascara. They can live up to 5 days in mascara. That was a study done out of Poland. So yes, change it often, especially after you come in for an in-office deep cleaning of the eyelids, like a Zest and an Ilux or a Lipoflow or something, or an IPL. Definitely change your wet makeup at that point, because you're just recolonizing with 8-legged pests. Did you know that they don't have an anus? They literally die full of bleep.
Did you? I don't think not everyone heard that, or they may have been in shock. Would you like to say that again?
No.
Yeah. You did say they don't have any.
They don't have an anus.
Anus. Okay.
Yeah.
All right.
So
this is an ophthalmology meeting.
Yeah.
And GI too mixed in a little bit.
They just
all
eat
right.
and breathe. That's it.
So what is your response when patients ask about over-the-counter lash serums outside of Lattice? Latisse. Is it fair to say just stick with Latisse?
Oh, that's such a good question. At least Latisse is fair, balanced, and transparent in the risks. That's what I appreciate about it. It's been studied for hypertrichosis. They are transparent about the risks associated with prostaglandins as a class. So if you're going to prescribe something or recommend something, at least it's FDA approved, and you've got that cover not only for yourself, medical legally, but also that level of confidence for your patient. Awesome question. Okay, last question. If you detect Demodex, and we all look for it, patients, you ask the patients to look down, and you see at the base of the lashes, the scurf, and if you detected, what are you using right now? Are you using tea tree oil? Are you recommending that, or hypochlor? Great question. So we do a lot of microscopic pulling too, because some patients are so good with their lid hygiene, you don't see them, the collarettes. So I'll do actually epilate lashes in suspected patients. Pretty much 50 to 60% of your MGD patients are going to have Demodex. So I do pluck a lot of lashes. I scan across the bridge of the nose for something I've described as the alpenglow sign. Follow my Instagram. There's some more details on that. So we do a deep clean. We do a Zest. And it's like really crunchy when you get started, because there's just like having your teeth cleaned. It's really satisfying. Get that stuff in there until you start getting smooth. Flip it over. Get it clean. Get after it, right? And then we teach them on home hygiene. We follow it with IPL. And sometimes if there's a really heavy mite load, I'll do a topical compounded ivermectin cream.
Okay.
Yeah.
Fantastic. Thank you so much for this very eye-opening, shocking lecture, as always. I appreciate
I took
it.
the assignment seriously.
Thank you. Thank you again, Laura. Laura came all the way from Seattle. What was that? Oh, another, I'm sorry, question, yes.
Do you have any recommendations how to clean lash extensions?
Oh, that's a good one. Come on back.
I have a question.
Just please clean them. Like, they get really gnarly really quick, like a camping trip and you forgot to pack an extra pair of underpants. So yeah, you gotta clean them. That was a traumatic experience when I was a child. But I'm working on formulating a foam-based cleanser specifically for eyelash extensions to minimize the breakdown of the glues because they're expensive, but yet get a nice, thorough clean. So I'm working on something.
Question. Yeah.
For now, I'm really reluctant to name brands. I'm so sorry, because this is a CE event. If you come up to me afterwards, I'll tell you what I'm using. It's an ophthalmologist out in New York who has a lovely foaming cleanser. So I'll talk to you about it offline.
All right.
Question.
Yes.
One last question.
One more question.
Yeah, question.
Oh.
Okay, OcuSoft makes a wipe for Demodex. Any opinion?
What's the concentration of tea tree oil?
I don't know.
You got to be 5% for 15 minutes to kill them.
Okay, when I get home,
Yeah,
I'll
so it
look
does a
at
great job at the bacteria, because remember how I said they don't pass feces ever, but their bodies degrade and spill it everywhere. So I think from the bacterial load perspective associated with Demodex, the wipes are great. Yep, that's appropriate thing, but you need much higher concentration to actually kill them.
The good news about Demodex is that there is a medication that's about to be approved, and it really is, it's a game changer. So be on the lookout for that. TP3, what is it going to be, the brand name? We don't know.
It doesn't have a name yet.
Doesn't have a
but,
name yet.
low to